password with you, the recording session. The recording session. So are you people able to download that recording or no?
I haven't tried to download, so I don't know.
Anyone tried if they are able to download?
No,
Because
I
I
tried, but
think,
not able to download.
yeah, because I think you won't be able to download.
Yes.
So that's the reason being asked. Now, today I got a message from Zoom that the storage is getting full. So that's why I asked whether you people are able to download, then otherwise I'll have to increase the storage amount. No problem. Fine. We'll see how to go with that. Okay, chalo. So your question was like for generic, what happens? Suppose if there is, because they have a defined reporting interval for everything, based on certain criteria are there, so that I can discuss some days later as to how we decide that reporting criteria. So what happens, suppose if molecule is there, which is having large number of ICSR cases being reported yearly. So what they will do that they will say that, okay, this is having large number of ICSR. So we will do its signal detection every month. So frequency for that is decided as every month. Okay. So now suppose in a month, what will happen? Like suppose 1st December to 31st December is its reporting period. So a line listing will be downloaded for that molecule, whatever events are reported in that. Now suppose if there are many drug event pair, like in 1 month, we got some around 200 cases. And suppose there are at least in that 200 cases, we have very commonly reported drug event pair at least 10. So what happens if it is achievable by you, means a single signal evaluation person, then it will be given to him, that only signal, that only drug will be provided to him to evaluate in that month. If it is more than one drug, means if it is not achievable by him, then it could be divided into two of them. So two people will work on the detection part of that, the whole line listing. So this is how it is managed. Okay. So it won't happen that if it is large data set, then usually what happens in a month, we get around 2 to 3 molecules are with one person. So they can work on that. If it is a complex molecule or more number of cases, then he will be given with only one drug so that he can work on that detection part. So this is how it is managed, where
So
we
it
are
is
doing manual.
achievable for one molecule,
Yeah,
all
yeah, it
the
is.
ICSR.
Yeah, it is achievable. We can do it.
Okay, so my next question is, so in the quantitative system, so there are some reporting ratios, or if
Mhm.
threshold is crossed, then there is a flag.
Mhm.
But if we are doing only qualitative assessments,
Mhm.
so how we will know, like, what is the threshold that it
No,
is
no.
a signal?
Yeah. So see, here you need to improvise a little bit on your concept. That threshold increase is just a criteria to flag a signal. Okay, that is not a signal, you can say. That is not a true signal from the quantitative, you can say. You mean, you understand what I'm saying? Like suppose some drug event pair is uh, passing the threshold. Okay, like suppose when you are measuring your blood pressure, it is 120 by 80. So if it is increased from that, then that is the threshold 120 by 80. That doesn't mean that if you get 130 by 90, then you have an increased blood pressure or you have a high blood pressure, right? So in the same manner, the threshold is 2.0. So quantitative signal detection help us to, without manual intervention, they help us to identify that there is a flag for signal, you need to evaluate it. Okay.
So there is a pattern or something, trend or something.
There is something that is fishy, you can say, that you need to evaluate. There is something change in the drug safety profile looking from the events being reported. So that is just a helpful statistical tool for you to not to analyze everything, because it happens there are many drugs in which we get thousands of cases in the reporting period. So in that case, to evaluate 1,000 cases, it is quite not possible. So that's why we keep that statistical quantitative one, so that once we get the flag, then we try to evaluate and validate that drug event pair. Okay.
Okay,
Both are part
understood.
of signal
Yeah.
detection only. Whether you are doing it through quantitative or you are doing it through qualitative, both are the signal detection process only. You are just detecting a signal. After that, you have to do a lot of things. But these both are for just detecting a signal.
Okay. So in earlier the signal session which we have conducted in our batch, in that we have done the categorization in detection part only, right?
Detection
But
part you can do. Detection part, because that was detection and validation both covered in that.
so in both the first in the detection part also we have to categorize, and then also in validation also we will categorize.
Yesterday also I told you that qualitative signal detection and signal validation both are same. The only difference is in qualitative you are analyzing multiple drug event pairs, and in validation you are analyzing only one drug event pair.
And the interval will be cumulative for validation and for the interval reporting period for detection.
Yeah, that is a difference.
Okay, that's what my question was. Thank you.
Any other question, people? No questions. So shall we move forward now today? Already we are running late. Okay. Chalo then. Today we are going to discuss mainly those sections of the PBERS which are basically we are not, you can say, directly responsible like we do for safety sections. The clinical trial team would be responsible for that, or the non-interventional team would be responsible for that. Most of the sections today are like that only. But what information goes in that we will see. Okay. So let's start first with the section 6. Okay. So what is the heading for section 6? Just a minute. Let me share my screen. I am not in a mood to write today, thinking that it will take time and I have to close the session early. Let's see. Can you see my screen?
Yes.
Okay. And most of the people are absent also today. Hello. We'll start with section 6. Tell me, guys, what is section 6?
Data and summary
Data and summary
tabulation.
tabulation.
Okay, so what do you understand by data in summary tabulation? Sorry. What do you understand by tabulations, summary tabulations? Any idea? Actually, summary tabulations, these are generated as you do line listing from your database. So these are also generated from your database, summary tabulations. So these are basically, you can say, auto-generated part from the database, and that we present into our as an appendix into our report. Okay. So the section 6 is, don't consider it as 5, it is 6 as per PBER. So section 6 is basically data in summary tabulations. Now what happens? The first section would be in this reference information you have to give, right? Reference information, what could be the reference here? People tell me, what could be the reference since we are providing data in summary tabulations? What could be the reference? Any idea? Will it be the RSI, the CDS or the RSI which you are using? Tell me. Any thoughts? What summary tabulation we are providing here? What would be the summary tabulation here? So it will be AE and ADRs that we are providing, right? Correct?
Yes.
So what should be the reference here?
Medra.
Correct. Using, you can write MEDRA terminology What version that you can mention? Version, suppose currently we are having 27.1, I think, right? So this is the reference how you will write. Why we are giving Medra version reference? Because we are giving the summary tabulations for AE and ADRs, and those AE and ADRs, how you will, how they are coded into the database, those are based on PTs, hierarchy of the Medras, PTs, HLTs, HGLTs, SOCs. So the AE ADR follow this hierarchy. So that's why we are giving which Medra version was used for this, right? Am I correct?
Yes.
Are you getting my point?
Yes.
Okay. Then we will go to section 5.2. So today's session is basically we are trying to cover. Mostly I told you that these are given by the people who are responsible for it. And certain things will be, you can say.
So, Amit, in 5.1, this is the only information, like we will give the reference like it is Medra version. That's
Boss,
it.
that's it. That's it. That is the reference information. Its heading is itself reference information. Okay. Shall I write a short form? SAEs. Okay. From clinical trials. Why only serious adverse events from clinical trial?
Because non-serious are not in database, reported in
Correct.
database.
Correct. That's why for these we consider only serious adverse events. Okay. So here what will happen that you will get. So your clinical trial team, they will provide you a summary tabulation for these for this section. So what you have to give here, there is a standard template statement for this, that a like it will be like a cumulative summary tabulation is provided. You can write it here so that you can remember that cumulative already is provided in the above. So they will provide you whatever cumulative summary tabulation is there for from the clinical trials, right from the DIBD. Okay. So this is a form of, means you can say appendix that you will get and you have to append to your P-bar. You will mention here that there is appendix 5.2 or 5.3, whatever appendix is there, you will mention here. And there is a standard template statement for this section, which we use. We don't utilize your, means you won't utilize your writing skills here. There is a standard template. I can tell you like how the template reads here. So that happens ki like that will say that a cumulative summary tabulation, suppose of all say SAEs reported in so and so clinical trials for the molecule from this date to this date is provided in so and so appendix, whatever appendix is there, and that is a ready to drop in section. So you no need to worry about that. You don't have to write anything. Are you getting it? What I'm trying to tell you here?
So the standard template statement will be there.
Yeah, standard template
You
statement.
have to change the dates if dates are provided.
Correct. Dates will be there because you know what the DLP for this molecule report will be changed. DIBD will be same. No need to change that. Then there will sometimes what happen, certain companies, what they do that they will provide as to how many size were reported here during the cumulative interval, and then during the reporting interval they will provide the numbers here only, and rest they will say that they will refer to the appendix, whatever is there in the appendix. What happens now, they will give you SOC and PT wise bifurcation. Like suppose if it is an investigation SOC, then the heading will be investigations SOC, and then in that whatever PTs are reported in that investigation SO under that investigation SOC, those will be there, and the number of cases reported for those PTs will be there. So this is how that appendix look like. Okay. So if anybody asks you, you can tell them that in 5.2 we provide a cumulative summary tabulation of serious adverse events from clinical trials, and that is attached as an appendix and is a ready to drop in section from the stats team or in clinical trial team we get, and that is attached. Okay. Fine, people.
Yes.
Next section. Shall I move forward, people?
Hi, Amit. Amit,
Yeah, tell me.
in here, section 5.2, you say, I mean, only serious cases are collated. So what about non-serious cases? I mean,
From clinical trials, we don't report non-serious cases. From clinical trials, only size are reported. Have you done the clinical trial ICSRs? Any chance?
yes.
So in that, you would have find only serious clinical trial cases
Yes.
that we present here. Means you're databased in your ICSRs, right?
Yes.
Yeah, but
So,
Amit, there are also ASIs which are non-serious. We report
Yes.
those also.
so that comes under the criteria of AESI, adverse event under special monitoring, or a special, you can say AESM, you can say, right?
So that will not be in this section.
That is not a part of clinical trial here. That will come under the below section, since post-marketing that will come now.
Okay.
Hello, Amit. I think it should be 6.1 or 6.2, right?
Your voice is not clear, Abhi.
I think it's 6.1 and 6.2. Na? You have wrongly mentioned 5.1. 6.1 is reference information. I'm not wrong.
Yeah, here, because in this section, see, it is automatically populating now. So earlier we wrote somewhere section 4. It is populating section based on certain in the Word document. If you will see, there would be some section 4 that is automatically taken. See, this we have given here. After that, it is taking section 5 there. Okay, so it is 6 only. Don't worry.
Okay.
I will write so that you don't get confused. Okay.
So, Amit, I process these city cases from ongoing trials. Actually, they are reported by investigators. So, in that, sometime there is a set criteria defined for ASI in protocol, right? It is ongoing. So, if that AE occurs in the patient, they also report that, saying that is an ASI non-serious event. So, we
I
do
think
submit those. Yeah.
in this case, if you can say, so do you mark IME in this case? No,
IME means
a important medical event.
no.
No, so it will be only non-serious, right?
Non-serious, right? But we will
Okay.
mention in narrative that it is an ASI.
Okay. So what happens now in this case? What is your question then?
So I am asking whether this information about this age that occurred, it will go in any section in paper.
Yes, it goes now where you are. See, you earlier we studied about the safety under monitoring, right? So your AESM, as there are different terms. So
ASI.
yeah, so you say AESI, somebody says a adverse event under special monitoring. Okay. So those cases will be presented in that section of 15.2. Like suppose if it is an ASM, suppose drug-induced liver injury is your ASM, and that is under your PSUR issues under safety monitoring as drug-induced liver injury. So when you will pull the cases for drug-induced liver injury, then it will pull that case, whether it is non-serious, serious, because that is a part of ASM.
But that is, if I'm not mistaken, it is for post-marketing cases, right?
No, there is nothing like post-marketing. If it is a clinical trial, then also it will be pulled. The criteria will be drug-induced liver injury, not the clinical trial or post-marketing. So that case you will present in 15.2 under special monitoring. Correct? Doubt?
No, I got it. So here only serious events will be pulled.
From clinical trial.
Yes.
This is section specific for serious adverse events from clinical trial. Okay.
Also, so I have one more doubt. So there are some countries for which they have obligations to report non-serious events as well for clinical trials. So those cases also, what we will do with those cases?
Suppose if there is some obligation to present some non-serious cases, then that will not come under this section. That is for sure. There will be only serious adverse events from clinical trials. But we will have to look, suppose if it is some local region specific, like let's see. Give me an example for Canada. We have a
China,
criteria.
China.
For Canada, let me say, for Canada, we have to present lack of efficacy cases. Okay. Like in PBER, you have the section that lack of efficacy from control clinical trials. But from post-marketing, we don't have any section for PBER, in PBER, right? But Canada says that they have to get the cases, they need to see the cases which have reported lack of efficacy from post-marketing also. So in that case, what we do for Canada local PBER, we have created we create a subsection in section 9, maybe probably 9.3 as a lack of efficacy. We will pull the post-marketing cases of lack of efficacy line through line listing, and we will present the analysis in that section. So in the same manner, suppose if China says that they need the non-serious cases to be submitted or to be reported. So from ICSR you are doing, if they have, as per the Chinese guidelines, if they are mandated that non-serious clinical trials cases should also come in the PBER, then you will create another subsection here, 5.2, you will create 5.3, or you can say cumulative 5.2 only as cumulative summary tabulation of size and non-size or of age from clinical trials. In this case, you will present the tabulation as you are presenting for 5.3, we will 6.3, which we will see in next section. So you can modulate it, because I told you that guidance documents are for our guidance only, though guidelines are there. But we can modify the as per the requirement of the health authority. So in this Chinese case, you can present it like that. No problem with that.
Okay, okay. Thank you so much for clarification.
So now in next section, you will see that we present both non-serious and serious because it is a post-marketing section. So same is there. You can say for post-marketing, I will write here. Next section is from post-marketing. Okay. So cumulative and interval. Here it will become like this. We will remove size from, you can say, PMS data sources. Or I should write. See, now here what will happen, you will present both the summary tabulation of serious, non-serious, and from the, and the same you will attach as an appendix. Okay. And if in this, we provide two type of bifurcation. For clinical trials, it is simply clinical trials and whatever serious adverse event. In this, what will happen that you will have bifurcation on the, like if it is a spontaneous report, if we will provide bifurcation by this spontaneous report, regulatory authority reports, literature reports. This one part from PMS, then second part will be solicited reports. Solicited reports you people have processed. You remember solicited reports, non-interventional studies, observational studies, those are solicited reports, correct?
Yes.
So now you have to have a catch here. When you are presenting the appendix in two parts, one is PMS part. So PMS part first will include the bifurcation based on spontaneous, regulatory, and literature. These are the main cases that we have to present here. And serious, non-serious, all reported from this. Okay. Let me draw a table, then you people will be able to understand. Typical table. Suppose you will write post-marketing source. Okay, post-marketing. Then you will bifurcate spontaneous. See how this will look so that you have an idea. Literature. Okay. Now what should I do?
Association and PTs.
Just a minute. I'll do it. Don't worry. Okay. SOC and PT also required, right? So Then the PT is in this SOC. Okay. Spontaneous regulatory. Oh. See, here you will get serious, non-serious. Same way it will be here. Okay. So now here it will be solicited. Okay. Solicited, what will happen? You will have only serious cases from solicited because that is a type of clinical study, you can say, non-interventional study. Okay. After that, you will have a column of
Sir, in 6.2, we will only take intervention study for clinical trial, right?
interventional only.
Okay.
To the right. Okay. One more column you can insert. Now what will happen? Just to give you a look and feel of that, I'm writing like this. See, here will be cumulative count. Cumulative, and then here will be interval count. Okay. So why it has come here? Need to demerge this. Why I'm not getting the option to demerge? This will be demerged because this is a part of literature here. We can merge it. So this is literature, okay? This is solicited only serious cases, okay? So this is how that looks like. So now if you have SOC, suppose investigation, so it will flag first investigation and then whatever like AST report, AST increased or where bilirubin increased, like those PTs will be there. So PTs and then how many serious cases from the spontaneous source, how many non-serious cases from spontaneous source, how many regulatory cases, how many serious non-serious regulatory cases, how many serious non-serious literature cases. If any serious case reported from solicited, then that will come here and total count will come as to how many were cumulative, how many were interval. So there will be bifurcation based on that only. So this is how this appendix looks like. Okay. So these are just the numbers that will be here and total number will be provided as to how many cumulative cases were reported and how many interval cases were reported. Okay.
So this bifurcation will already be done by that team, right?
Yeah, that will be from the SAS teams only. They will provide you a ready to drop in. You don't need to do anything. See, in many generic companies, what happened now? They provide you, they ask you to generate a cumulative line listing, interval line listing separately in this format and then merge them and provide a single appendix for that. So, but that happens in certain generic companies. Like I told you yesterday, now there are many generic companies we don't invest in their tools for Signal. So in the same fashion, they don't invest much in all these things. They ask their people to do that. But most widely, 95% you will find that it is auto-generated from the system.
Amit, so all type of reports you have mentioned, there are patient support program or market research programs reports also. So in which category they will fall into?
Patient support program goes into non-interventional, right?
Yes.
Market research will also go into non-interventional, I believe, solicited.
So, but
So they will
they are not for the adverse event, what you can say, not for the sake of adverse event, collection of adverse event. They are just supporting the patient, right?
Yeah, but they are the solicited programs, na? Means sponsored by your company in which you are directly not involved in medical intervention, but you are monitoring those things, right? So if person is, why patient support program is there to support the patient and also to monitor your drug also, right? So if some patient is taking medicine from a PSP and they come and report, then only you will get that PSP report. So there is a criteria to check in ICSR as a solicited, those PSP reports. So you will check it as solicited and it will definitely fall into this category when we are generating the line listing, right?
Same goes for the individual patient use and compassionate.
Yeah, whatever is you are, see, when you are generating a line listing, so what is the convention for that compassionate use? Do you make it as solicited? How is it?
Yes.
Or other clinical trials? How do you make it?
Solid in non-interventional studies.
So that will come under solicited. Whatever you have checked in ICSR, that will come under here, solicited. That's why a convention is there in ICSR to categorize the case, right?
Right.
To facilitate these processes only. Done? This section?
Yes. Amit, one more question. Why we are not taking interval data for clinical trials, only cumulative? Hello, can you hear me, Amit? Other guys, can you hear me? Anyone?
Yes, sure.
Yeah. Can you hear Amit?
No, we can't hear.
Amit, we cannot hear you.
Hello? Can you hear me now?
Yes, yes, we can hear you
Yes,
now.
now.
Can you hear me, people? Hello?
Yes, yes.
Yes, Amit, now audible.
Now audible?
Yes.
Okay. Sorry, I think there was some mistake.
So
So
I
what
did
is
not
this
hear your answer actually
answer
about
for
cumulative data. Cumulative data, why we are presenting only cumulative data in clinical trials?
only cumulative data where?
In 5.2, the 6.2, clinical trials, the summary tabulation.
Here?
Yes, cumulative
Yeah.
summary tabulation for SUV.
So in this, it is not necessary that you have to provide only cumulative. You can provide as summary, means that interval also. But what happens in the clinical trials, we don't have enough data. Means you cannot say that there are many size reported. That's why we keep it as cumulative, so that everything comes under one, means one bucket, so that we can You can say for clinical trials, we can analyze cumulatively as to what size were reported. You won't find many size reported in clinical trials. That's why we keep it as cumulative. Why we have bifurcated in post-marketing? Because we have many cases that we get in reporting interval also. So we need to bifurcate that there is these many cases reported in interval and these are the total cases reported till date. So that's it. There is nothing specific reason why we are not. You can represent as interval bifurcation also in clinical trials too. But that quantity will be very less. You can do that. No, there is no bond, means there is no hard and fast rule that you have to do like this only. Okay. Is it clear?
Yes, Amit, thank you.
Yeah. So I was asking like section 7, what is the heading? By this time you people should remember as to what is the heading. Because somebody will directly ask you what do you present in 7.1. At that time you will just ponder as to what was the heading for that. So try to remember the headings at least what you are doing for papers. Okay. Chalo, tell me what is the heading for section 7?
Summary
Tell me.
significant
From?
finding from
Significant finding in the city.
From city. Okay.
during reporting interval.
During the voting period. Correct?
Yes.
So in this, what will happen? What will happen in this? I could have told to some of the people who were there. Any idea? What could be there in this?
Any risk identified during a clinical trial of their drug?
Not the risk.
So whatever studies are completed and ongoing studies are there, we will present them.
Then why do we say significant safety findings?
Those are not finding in the previous studies that we are finding in here.
Any other thoughts? Tell me. So see. Now first section in this will be 7.1. What is this? That clinical trials. What could be it? Clinical trials completed, right? During the reporting period. Okay. So now you will say, like, suppose you say that there was one clinical trial completed. So first of all, tell me what is the definition for clinical trial completed? During the reporting period, like you say that one company sponsored study was completed. Sponsored study, then you can give study number. Study number was completed. What is the definition? Tell me, guys. Don't keep quiet.
When it is signed, stamped, and approved by appropriate.
Yes. When
What?
CSR is signed, when CSR is signed and approved.
Correct. So if the CSR is stamped and approved, means it is signed and approved, then for that clinical study, you will say that it is completed. And you will provide a tabular format here. The tabular format will be like the same that we discussed. No need to draw, I think. You will provide the clinical study protocol number. You will provide what was the title of that protocol. Then you can provide as to when was the last date. I think for this, no. For completed, we don't need to provide the last date of last patient visit. And then if there is any clinically important findings or safety findings that you have observed, based on the CSR that you will present here. So it will be tabular format and short form. So mainly what happens that this section is provided by the clinical trial team, but you can also write this section and ask them to endorse it, whether you have presented the data correctly. So this is basically the clinical trial team, clinical science person responsibility. But you can populate and ask them for confirmation. Okay. So what do we provide? What information we provide in this section? Anyone? What I told you.
Whatever significant information.
No, whether there is significant information or not, but you will provide, like you said, now one company sponsored study was completed. So what you will do here, you will mention that study protocol number, protocol title. Protocol number is the same that is here, study number. So protocol number, protocol title, why this study was conducted, all those things in protocol title and its purpose. And then you can provide if there was any significant safety finding. If there was any significant safety finding that you can see in CSR, you can list here. Otherwise, you can say that no significant safety finding was observed in this study. Simply. Correct?
So in CSR, they will mention it like significant safety findings. It will be titled.
No. In CSR, there will be a safety section, there will be efficacy section. So in safety section, you have to see whether there is any significant safety finding or whether there was any safety finding which changed into signal which was evaluated. So these kind of things you need to find in the CSR. It will be presented there. That's why I said now that is the responsibility for clinical science team, but you can populate and ask them to confirmation. If in the interview you are saying that this is the data that we present here, however, this section is dropped in by the clinical science team, it is fine. Nobody will have question. Okay.
So
Okay.
this is study wise, right? Like for every clinical trial we have to do this.
Right. For
Mm.
every clinical trial, which is completed during this reporting period.
Okay. So in section 6.2 where we present the essays, there also we have to go by every study wise, or we will add all the data in one tabulation.
No, whatever clinical trials are going or whatever cases are reported in clinical trial safety database, for all the clinical trials, it will be there. Some might say that we need bifurcation on study wise, so they can get the study wise also. It will be there. Study number will be mentioned there. So there is nothing thumb rule. What suits your need, our sponsors need, they will ask to follow those conventions. But one thing is for sure that you should not miss any safety finding to be reported to health authority, either via your own management system or via PBOR or something. You have to report everything that is significant. Okay. Any further question, people? See, these clinical trial sections are very confusing if you don't get the concept properly. So try to understand everything. And one more thing is there that these sections are not drafted by us. Most of them are either ready to drop in or the clinical science responsibility. So that's why people ask these questions in interview, because they want to know whether you understand or not. It's fine that it's somebody else's responsibility, but whether you understand what you're writing in the paper or what you're presenting in the paper. Okay. Chalo. Shall we move forward?
Yes.
7.2. Yeah, tell me.
In section 6 also, we provide this all tabular form in appendix, and also we write the summary in written print in whatever template is there, right?
If your client asks to write the summary in this section, so summary will be only numbers. That like suppose if you say that there were totally in cumulative interval you received 1000 cases, out of which they will say that bifurcate based on source. So you will say that there were 900 spontaneous cases, 50 literature cases, 20 regulatory cases, and suppose 10 from solicited. So this bifurcation you will give. Also, you can give there was these many serious cases, these were non-serious cases. Simple, this one. Nothing else apart from that. No summary as to what was the safety finding, nothing. Just the numbers is the summary that you provide because you are attaching appendix here.
Also, we will mention the SOC and PTs, how many are serious number, right?
That we are providing here na, in the appendix. No need to mention in the summary section there, in the section above.
Okay.
Otherwise, what is the need to provide this appendix then?
Yes.
Okay.
Also, this section 7 is for reporting period and section 6.2 is for cumulative for every study.
Cumulative and interval both. Section 6 is there. Section 6 is having cumulative and interval both for post-marketing. For clinical trial, if you wish, your client says, then you can provide cumulative and interval. Otherwise, cumulative is fine.
Okay.
Okay. Now the next section is clinical trials ongoing during the reporting period. So suppose if you say that there were during the reporting period, suppose you say that there were 3 companies sponsored clinical trials. So you have to provide the study number for all 3 here. And then yeah, you can write were ongoing during the reporting period, and then the table, the same table as you provide above. The same title will be there, protocol number, protocol title. One thing you can add here in the table as to when is your last patient last visit expected, estimated date. That will be there in protocol as to when is the last patient last visit expected. So you can write that date here, estimated last subject, last patient last visit, or last subject last visit. You can say for clinical trials we don't say patient, we say subjects. Always remember, in clinical trials we say subjects. So last subject last visit, expected date you can provide here, and then one column for summary of significant safety findings or significant finding, where basically we say that clinically significant finding. So what will happen that you will provide, like if you are doing that clinical trial and for certain reason, means you are trying to investigate its efficacy in a certain indication and you are reaching that efficacy endpoint. So you are, suppose you want to present here, so you can say that it was found to be effective in so and so, and if there was any significant safety finding, that also you can present here. Both you can present here, but
Till
efficacy
DLP.
in the reporting period till DLP. Okay. Am I clear? People, am I clear?
Yes.
Shall we move forward?
Yes.
Okay.
Sir, also, can you give 4 to 5 examples? What significant findings we write about the trial?
Like suppose there was one clinical trial, suppose ongoing, and you found event signal in that. Suppose there was some, you can say a bleeding signal was observed in that. So that signal was evaluated and all those things. So if that signal was observed like bleeding, so the same you will present here that there was a signal generated from the trial, this bleeding. And if the evaluation is completed, you can provide its summary also that what happened to that. Means suppose if that bleeding was confirmed by you as a risk, then you can write that it was confirmed as a important potential risk or important identified risk and the RSI was changed for that. So in this way, you can write the significant safety findings. Okay. If evaluation is not complete, then you can simply write that the signal of bleeding was observed or that is still under evaluation. Or suppose if that is not a signal, if this is something important event that was observed, suppose there were 3 cases of thrombocytopenia. So in that case, you can write that there were treatment emergent adverse event observed which crossed the threshold, and that means which were reported. But you can say that there were 3 cases of thrombocytopenia reported as a treatment emergent adverse. So these are the language for those clinical trials: treatment emergent adverse event, size, and all those things. So once you go through some CSR or something, then you will understand those terms. So in this way, we write this. So that's why I'm not explaining all those things, because that will be more confusing for you people. So these are the significant safety findings that you get present here. Okay.
Also,
Shahrukh, you can continue.
so Amit, this request we will give to the clinical trial team by minus DLP minus 80 days, right?
Or what?
For all the information we
No,
need.
they will not provide you information like this. They will provide you whatever outputs you ask them. Okay. Like you will ask them the above output appendix, that cumulative and that section 6 outputs. So these outputs they will provide, or your post marketing statistical team will provide all these outputs. What will happen when you will initiate this document for collaborative authoring? Okay. Because whole paper we don't write. We are safety writers. Okay. We will write the safety sections. So what happens? First, we write the safety sections, and then we initiate the collaborative authoring for them, for these sections, like this one, clinical sections. So in the system, if you have populated certain data, they will either confirm it, or they will provide their data in the system.
So by which date, according to DLP?
You can initiate it pre DLP itself. Means by DLP, you can say, suppose you want to end the authoring by DLP plus 30. So you can initiate the authoring by DLP minus 20 also. So DLP minus 25 also, you can initiate the authoring, collaborative authoring for other sections to be populated by other people who are responsible.
And it will be finalized DLP plus 30 days.
By that, authoring should be completed, and then reviews will start. Okay. Just a minute, guys. Give me a sec. Keep your questions. Yes, guys. Actually, today it is very hectic since morning itself. No problem. Tell me. Done? Shall we move forward? Okay. Then what is section 7.3? Anybody? Tell me.
Long-term therapy.
Long-term follow-up.
By this time, you should understand what are the section headings. Chalo, tell me, what would be there in this section? How can you identify that whether there should be some information in this section or not? Any idea?
So after the clinical trial is completed, whatever the safety findings we reported during long-term follow-up, we'll take it here.
Correct. What else could be there? How can you identify that we have to follow up on this long term?
So there are protocol specific long-term follow-up defined.
Correct. In the protocol itself, it will be defined that whether you have to do the long-term follow-up or not. Based on that, you will monitor. If you receive some information on long-term follow-up, some safety findings from long-term follow-up, then you can mention here. Otherwise, you can simply say that the subjects who completed are not subject to long-term follow-up. 2 criteria will be there. One, your protocol doesn't say to have a long-term follow-up. So in that case, you will write that the subjects who have completed the study are not meant, or you can say are not subject to long-term follow-up. So that is because your protocol doesn't say to have. But if your protocol says that you have to do the long-term follow-up and you don't find any safety finding, then you can present here that there were no long-term follow-up safety issues identified during the reporting period. If you find a safety finding from the long-term follow-up as per protocol says you have to follow, then you can mention that safety finding here, that this was the safety finding observed in the patient, in the subject on long-term follow-up. Whatever information is there, like post 6 months of drug exposure, the patient observed to experience this event. So you can present in this format. Okay. Just tell me.
So how this will be presented, same as ongoing and completed?
This is in the paragraph format.
Okay.
Okay. You will mention the study name, that this study name has a long-term follow-up in which the subject observed this after these many days of exposure. You have to provide how many days of exposure was there. So you will have to provide after these many days of exposure, the patient subject experience this adverse event in so and so study. ठीक है?
Yes.
So what is the next section? Tell me. Next section.
Other therapeutic uses.
Other therapeutic use of medicine or product.
So what could be the other therapeutic use? Can you tell me, can you give me an example? What could be the other therapeutic use?
Compassionate use.
Correct. What else? So whatever is expanded access program, whatever is compassionate use, but that should be sponsored by your company. It should not happen that somebody is doing on his own and you are reporting it. It should be sponsored by your company. Sometimes it happens like, see, there is a scenario like suppose if I am investigating my molecule for certain indication in a certain region, let's say UK, and my drug is approved for the same indication is EU. So now I am still investigating its efficacy in UK region. Okay. And I have to apply for it. So now what will happen before my approval, if I am giving my medicine as a sponsor to the investigator or to certain prescribers to prescribe it in the same indication. So these are called expanded access or compassionate use programs. So since you are only providing as a sponsor, you are taking responsibility to provide that medicine. So you will present those kind of sponsored therapeutic use or compassionate use or expanded access, whatever safety findings you are observing from them, you need to present it here in this section of the PWAR. Okay. But it should be sponsored by you. Remember, means you means MAH.
So, Amit, any data from other trials which are supported, not sponsored but supported, or any other clinical trial which other companies are using our drug?
There are other, there is a section 9.9 information from other clinical trials and sources.
Yes,
In
yes.
that you will present that. Okay. Shall we move forward? And this is your responsibility, this section, 7.3, 7.4. 7.5. What is 7.5? Tell me, guys, what is 7.5?
It is for combination therapies. Any safety
Yes.
data?
Yes. So combination therapies, what do you understand by combination therapies? Tell me.
So, if two drugs are investigated simultaneously, like in a combination.
Yeah, like suppose you are doing the clinical trial for your molecule, like says, let's take an example of suppose azithromycin only. And if azithromycin is also being uh, marketed in a combination, means, for example, let's say you do not azithromycin. Let's take a famous example of amoxicillin and clavulanic acid. So you are writing this paper for amoxicillin, but it is also marketed as amoxicillin and clavulanic acid. So if there is any safety finding from the use of that combination product, that is amoxicillin and clavulanic acid, so you will provide it here. In case if your drug is not into the combination product, you can straight away mention that this is not applicable because your drug is not in the any combination therapy. And if it is in the combination therapy and you find some safety finding, you can mention it here. If it is in the combination therapy and you are not you have not observed any safety finding, then you can write the negative statement that there was no significant safety findings observed from any of the combination therapies that the drug is being marketed.
So
Simply.
if the drug is not yet marketed, but it is under investigation on in combination therapy, then
That information will come here, na? If it is under investigation in the combination therapy and you are getting some safety finding from that clinical trial, then you will present here.
so from both post marketing and clinical trial will present.
Both. Okay.
Only for approved, right? Whatever.
Whatever you are approved combination therapy or the combination therapy which is under investigation. It should not be like that. Suppose you are marketing azithromycin and somebody is giving azithromycin and then one more tablet of something like that, some other like paracetamol. So you won't consider it as combination. It should be in the same formulation. Okay.
Yeah.
Any doubt?
So Amit, if the drug is not marketed individually or investigated individually, but itself it is a combination therapy. So that also will. So like suppose
No.
two drugs are there, and
So,
yeah,
so tell me, will you write this paper for combination therapy then or for single molecule?
for individual molecule.
Why?
मतलब three papers we will write for both the molecules and one is for
No, you mentioned that your drug is being marketed in a combination therapy only. Then why
Yes.
will you write individual paper? You will write the combination paper only.
Combination paper. But
Correct.
in that guideline, it is saying that we have to write individually for
No,
both the molecules. Safety
no,
finding.
no. See, now I'll tell you the scenario, what will happen. Like suppose there is coamoxiclav that we were asking about, like amoxicillin and clavulanic acid. Now suppose if you are a MAH for amoxicillin as individual, clavulanic acid as individual, and also an MAH for coamoxiclav. In this condition, you will write three papers. But if you are only the MAH for coamoxiclav, then in this condition, you have to write paper only for coamoxiclav. That is combination product and not the individual. Got it?
Yes, yes, I understand. But shall I read the sentence
Yes.
from the guide? So
Yes.
if the product itself is a fixed combination product, this PSCR subsection should summarize important safety information arising from the individual components, whether authorized or under development.
Okay, so you are saying that
Mhm.
when you are writing a paper for a combination product, okay?
Yes.
Am I correct? When you are writing
Yes,
this
yes.
this paper is not for single product. This paper is for combination product. In this case, new safety data related to combination therapies. In this section, you are saying that you will present the individual data, right? Like
Yes, yes, that's
co-amoxiclav is
one.
there.
Yes.
So you will have, if you have any finding from amoxicillin or clavulanic acid, then you can present here, right?
Right.
So in that case also the same is applicable. You should be MAH for amoxicillin or clavulanic. Then only you will get the cases and information, right? Otherwise, how will you get that information?
Yeah, but those cases will have information of both the drugs. Like together they are causing some adverse event. So how can we differentiate that they for this drug this is the adverse event and for this drug this is the adverse event?
You are confusing yourself. See, there is a scenario like suppose you received a case, okay, in which co-amoxiclav is the suspect. There is one scenario in one case, okay? Got it? Co-amoxiclav
Yes,
means a combination of amoxicillin and clavulanic acid is a suspect in that. Okay.
correct.
There is another scenario, a case where only amoxicillin is taken and that is the suspect.
Hmm.
And then
Right.
third case where only clavulanic acid is taken and that is the suspect.
Okay.
Okay. So will you not be able to identify as to what is the suspect from which
Yes,
drug the
yes.
event is happened?
Yeah,
So
yeah. But that
suppose,
yeah. Mhm.
yeah, tell me.
In this case, MH is marketing individually as well as a
Combination.
combination, right?
Hmm.
But I'm saying specifically if it is marketing only as a combination,
Hmm.
then how can we differentiate individually for both the molecules?
Okay, tell me one thing. If you are marketing it for combination only, then if you get a case of amoxicillin only, will you database in your ICSR?
No.
Then how will you get that case? Then how will you
We will
get that
not.
case? When you are not an MAH, you will say that I am not an MAH. You will directly non-validate that case, right?
Mhm. Right.
So how
I
will you get
understand
that
that.
case?
Yeah, but here they are saying if the product itself is a combination product. So they are
Huh?
saying that it is a combination product. So for that combination product, we have to write it separate for each molecule.
What separate? That I'm not able to understand what you are saying me to write that separate.
Yes, Sato.
Separate
This is
paper.
7 point. Yeah, no. The 7.5 section, new
Uh,
safety data related to the fixed combination therapy. In that, they are saying unless otherwise specified by national or regional regulatory requirement, the following option can be used to present data for the combination therapy. If the product itself is a fixed combination product, this PSUR subsection should summarize important safety information arising from the individual components, whether authorized or under development.
So if you are authorized or if you are doing clinical trial, then only you can present. How will you get that information? That is my question. If you are not an authorized person, means authorized MAH for that single molecule, how will you get that case to present in this section? That is my question.
Allow me,
Are
sir.
you trying to understand? Like suppose, if you are only authorized to market amoxicillin and clavulanic combination, not the individual, either amoxicillin or clavulanic acid. Okay. So if you will get the case of amoxicillin or clavulanic acid as individual, you will non-validate that case. You won't database into your ICSR, right? If you are only
Mhm.
MAH
Right.
for combination.
Mhm.
So you
Right,
will not
right.
database it. But in case if you are MAH for all the three types, mean the combination also and both the individual molecules. In this case, if you are writing a paper for combination product that is co-amoxiclav, in that case, in this section in 7.5, you will
Mhm.
write the information or significant safety findings that you are finding from individual molecules, whether it is from amoxicillin or whether it is from clavulanic acid. But you should be MAH for those.
Mhm. Right. But I understand that. So now the question is why we are incorporating that individual component information in combination product paper.
This therapy because you are writing a combination product paper na this is not an
Mhm.
individual product. See,
Yes.
if you are writing a combination, if you are writing a combination product, then both molecules are a separate entity. Okay. So
Yes.
what would be the combination product in that in this 7.5 when you are writing it for combination? Suppose co-amoxiclav paper you are writing. So
Mm-hmm.
that is itself a combination therapy. Okay.
Yes.
So in this case, what will happen if you are an MH for component also, and if you are getting any significant safety finding from that, you will present in section 7.5.
Mhm.
Okay.
Yes, yes, yes.
That is the scenario. That is the guidance, what it is saying, guideline.
Mhm.
But you should
So
be amazed for
basically,
it.
if it is a combination P-bar, then we will first mention the any significant safety finding from the combination product itself
No,
for
no,
both
no,
the.
no, 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 no. In this section, if you are writing for
Hmm.
combination product, then whole
Hmm.
PBR is for combination product. You are writing all the significant safety findings from that combination molecule in the whole PBR,
Correct.
all the sections. But in
Yes,
this
correct.
section, 7.5, you will write whatever is the significant finding from those individual molecules.
Mhm.
Okay?
Okay, okay. Hmm. Right, right.
Sure. Let's move forward.
Okay.
Section 8. What is it?
No, Amit sir, I have a
Tell
doubt.
me.
Means for example, if we're writing fever for a clavulanic acid, right? And if our product is a combination product,
मैच ला
amoxicillin
ला
and
के रखना
means clavulanic. So we will only write the data.
Sorry to stop you. Just be a bit loud and be clear. Start your question again.
Okay. I'm saying, see, if our product is single, we are writing for clavulanic acid, right?
Hmm.
And if it is given in combination like with as you mentioned with amoxicillin, right? So in this we will write only whatever is about clavulanic acid, right?
Where?
In this combination therapy.
No. If you are writing for clavulanic acid, a single molecule,
Yes.
then you will present in this section the significant safety finding from clavulanic acid and amoxicillin combination product.
Then we will for combination product we will write.
Yes.
Okay.
If you are
And so
writing
the
for
vice,
single molecule,
yeah, it is vice versa for if we are
combination
writing
product.
combination product, then we will mention individual here.
Yeah. If you have any significant finding from individual, you should be amazed for that. And you have any significant safety finding, you can provide it here.
Okay, understood.
Chalo, next section. Tell me guys, what is the next section?
The findings from non-interventional study.
Okay. Trial or studies. Okay. So it is also the same. Means you will have to provide the information about your non-interventional trial that you are doing. Non-interventional trial by this section, what do you mean? Because we are providing solicited information also above. But here we are saying that safety findings from non-interventional trial or studies. So what could be these non-interventional trial or studies? Any example can you give me?
Any observational study?
Any observational study? Yes, it can be.
So disease registry.
Registry could be there. There could be some non-interventional pass studies, post-authorization safety studies, like suppose in certain, like suppose if you have to observe certain disease patients specifically, like say, if you have designed a trial to monitor the myelofibrosis patients. So you will enroll all the myelofibrosis patients into your study and then you will give your molecule to them. It is a company sponsored non-interventional trial because you have taken approval, you have a protocol for all these things and you are authorized to do this. So you will enroll all the myelofibrosis patients into your trial and then you will observe whether they are having any safety findings or not. And if there is any safety findings, you can present it here. What is the purpose of this non-interventional pass studies? Basically, it is to characterize certain risk. Because like suppose if you say that myelofibrosis patients are more prone to have, you can say fibrosis. So fibrosis is a risk in myelofibrosis patient for you. In this case, when you want to characterize this risk, so you don't have enough information at its starting and you want to characterize this risk further for 16.4 or for RMP, you can say. So you can design a non-interventional pass studies in which you will enroll all the myelofibrosis patients and you will observe whether they are having fibrosis or not, or they are having some other safety issues. So these kind of things are presented here. So what you will present in this information in section that so and so study is being conducted. What is the name of the study? It will be in paragraph format. not tabular format necessary, or if some client says, then you can present in tabular format also, as you are doing in section 7.1, 7.2. So you can write like this so and so study, this protocol number, then what is the title of that, means what is the purpose of that study. And then if there is any significant safety finding, or how many patients were enrolled into that, then how many patients had that safety finding. So in this way, you can write a paragraph in this section. Okay.
So, Amit, any real world study we are doing, that is also we will include here?
Yes, real world study also sponsored by your company will come under this.
Okay. For general question regarding missing information, for if are we doing any past study for any missing information in risk?
Past studies for missing information, I haven't come across any. So I don't think that we plan any past studies for missing information. The reason I'll tell you, because we don't characterize the missing information. Okay. And past studies main objective is to characterize your risk. Basically, the main objective of past studies is to further characterize your risk. And in this case, this happens when you are doing something, when you are working for innovator molecule. And at that time, since suppose your molecule is into the market from the past 1 year, 2 year, there are certain risks that came from clinical trials, and that risks are not characterized properly. Means you haven't seen much impact on public health, or you haven't observed any safety findings in the when it is into the post marketing setting. At that case, what do? We plan all these past studies, and we specifically monitor if there is certain anticipated risk. And as part of that risk minimization, also we keep these past studies. So that's why this is done for mainly important risk and not for missing information. Because missing information is not a risk, you can say. It is a gap, it is a knowledge gap. So that's why no past studies for missing information is required. Until and unless there is a scenario that, like, suppose your drug can be used in pregnant ladies. At that time, you have to monitor that. Make a past study or observational study in which you will you need to have you need to monitor whether any pregnant lady is taking or not. Other, for missing information, we don't plan any past studies. Have you come across any in your ICSR?
No, I was just asking, like, if later on, after the drug is established, we will remove some things from the missing information and categorize as a potential risk or important identify if we have enough data and knowledge. So why are we not doing any past studies earlier to categorize it as a...
No. Past studies are done to characterize, not to categorize. Okay. So missing information, no need to do characterization for missing information, because that is a knowledge gap. Characterization is required for your important risk, whether it is identified or potential. That's why we do past studies for them, and no past study for missing. No need to characterize missing information. Remember. And as you are saying that it can be removed from missing information to important potential risk, in that case, what happens? That is a upgradation of the risk, you can say. Okay. It is recategorized. Okay. Always remember there's a difference between categorization and characterization. Okay. So in this case, what happens? Suppose if your missing information is moving into important potential risk. So in this case, what will happen? Like suppose using pregnant ladies is your missing information. Now in certain reporting interval, or in you can say it would be suppose it is a 1 year reporting period. In that you started finding significant safety findings you have been observed. You are starting observing like teratogenicity itself in pregnant ladies you have started finding. So in that case, you will open a signal, you will evaluate the signal, and then if you confirm the signal as a risk, then you will categorize it as a risk, whether it is important, potential, important. This is the process that is all followed. And then if it is subcategorized as important risk, whether it is potential or identified, your RSI will be updated, your missing information will be updated, upgraded to important, potential, or identified, and then you can start past study for that, post that, once you have categorized it at risk.
So unless something is reported regarding missing information we are receiving, then only we will open the signal. Otherwise, we will not initiate from ourselves to research about that in missing information.
No, that's why we are monitoring that missing information. No? Until there is signal, we will keep it as missing information. Not only signal, until that signal is confirmed and changed into risk, we will keep it under missing information and monitor it into our PBER only. We will present the cases whatever we receive for that missing information, and we will analyze the cases and present our analysis and our conclusion on that. We will keep doing like this. And if at a certain interval, suppose after 4, 5 or 6 consecutive or 10 consecutive years, whatever is there, if we don't find any significant information in that missing information section, then we can remove the missing information stating the same.
Okay, got it.
Anything else, people? Done? Section 8 done? We have only this information from section 8. And then 9 to we discussed that day, na? 9.1 and 9.2 both, right? Right, people? Yes, no, maybe. Can anyone confirm?
So I think we have discussed 9.2 in details. I'm not
Not
sure
9.
about
9 is
9.1.
nothing, but it is the 9 section itself says that information from clinical trials and other clinical trials and other clinical trials and other sources, right?
Yes, information from other clinical trials and so.
Yeah. So other sources like medication error is presented in 9.2, and 9.1 what we have, that is other clinical trials. So suppose there is some investigator sponsored clinical trials ongoing, which means you are not directly sponsored it. Means as a MAH, we are not sponsoring it. Means we have not control on that. But there is some investigator initiated clinical trial. So in that case, what happens that an investigator might ask you as a sponsor to provide you the drug. So in that case, it becomes your liability to provide them the drug, but ask them to report any safety findings. So that party, that investigator will report you if there is any significant safety finding that you can present in section 9.1. Otherwise, you can say that there was no significant safety findings observed from any of the investigator-sponsored clinical trials. That is, you are not as an MAH sponsoring that. You are just providing the drug to that investigator who is doing that trial. Got it?
Yes, got it. So Amit, can you refresh from section 6 to section 9, which section are ready to drop in and which we need to collect the information and write?
See, from section 6 till here, you get ready to drop in or the contribution of clinical size. But from here onwards, you have to write whatever you get from a database. Okay.
And the source will be the clinical trial team.
No, your safety database could be there. Na? If there is any long-term follow-up, it will be in your post-marketing case. If there is other therapeutic use, it will be coded as compassionate use in your product database. Is combination therapy, this also you will get from your, it might happen that there would be some combination therapy under investigation. So in that case, your clinical trial team will provide. They are also in the collaborative authoring, they will provide it. This non-interventional trials, there is non-interventional team that will provide you the information which you can incorporate here. So most of the sections are ready to drop in or a stakeholder contribution is related, means required here from section 6 to 8. Okay. Clear?
Yes.
All other people are just quiet. They don't have any doubt. Actually, today's strength is also very less. That's why these recordings, people rely on recordings now. They are learning at their own pace. At least if this is the condition that most of you are not able to join, then we can postpone the session on this day. Chalo, koi nahi. What is the 10th section?
Non-clinical data.
Okay. So what information should give here?
The clinical study data.
Correct. Animal studies, you can say simply. Whatever animal studies are there, that will If any significant finding from animal studies, then you can present here only significant safety finding. If there is nothing, you can straight away mention that during the reporting period, no non-clinical studies with fed drug on tinib was. Either any study was ongoing, so ongoing or completed, or any significant safety finding was not observed. So you can write no new significant safety finding was observed from those studies which were being evaluated. Okay. In short, as you are presenting the clinical trials data during the reporting period, in the same way, non-clinical data also you can, if there is some animal study ongoing, you can present in tabular format that this was the animal study that was ongoing, what was the purpose of the study, and if any significant safety finding was there from that study. Simply. Okay.
The source, Amit, who will provide this information?
Non-clinical team is there. Toxicology team, pharmacology team, PKPD team, non-clinical team we say. They are the source for this.
I'm sure only for our reporting in internal, right? This is whatever you write in.
Correct. Only during the reporting period.
And this also we directly get from that team.
Non-clinical team, yes, they will provide you. But you should know as to what goes in that section. It's fine that they will provide, but in interview, somebody is asking, they are expecting you to know what goes in this section.
Yes. Also, Mr. Any other studies which go in this, like animal study?
No.
Other example.
Only animal studies, no human studies.
Okay.
So any in vitro or
That will go, yes.
In vitro, in vivo, and all.
Whatever you do in lab, that will go here. Not on human beings directly. Done?
Yes.
Chal. Then we'll move forward. What is next section?
literature?
Section 11.
Correct. So what information will go here? Any signal literature. What do you understand by signal literature? Suppose in this section, always remember, section 11 is basically for the information from the literature which is not present in your RSI. Okay. So ideally, you can say it could be a signal. It could be a literature for special population. Special population means any safety finding from that special population. Okay. Like suppose if it was not indicated for hepatic impaired patients, but this literature states that it was used in the hepatic impaired patient, and the patient observed some safety findings, means their liver enzymes were raised abnormally or something like that. And that special population is not part of your missing information. Okay. Like in this case, I mentioned that a special population literature in hepatic impaired patients. So that use in hepatic impaired patients or hepatic impaired should not be a missing information. You are not monitoring that under missing information. So in that case, for that special population, whatever study safety data you are finding in a literature, that kind of literature you can present here as a special population. Okay. What else could be there? Class effects. Class effects. Like suppose if you are using, let's say, omeprazole is your molecule for which you are writing this paper, and then you have a literature in which pantoprazole was used and it has certain safety finding, which is not part of your omeprazole RSI, that safety or ADR or a risk. So that kind of safety literature you can present in this section as a class effect. Okay. Am I clear? Any signal literature means if there is some information that is turning out to be a signal from that literature, like it is qualifying the criteria of signal, where either it is a new aspect in comparison to your RSI, or it is a new aspect of a known information in your RSI. So that kind of literature or a safety finding in that literature, that will be presented here. Second thing, special population. If that special population is not part of your missing information, Because if it is a missing information, then you will present that literature into the missing information. But if it is not part of your missing information, you are not monitoring it as a missing information, then that special population safety finding will come under this literature section. And then class effects also. So if you're like, I give you the example of omeprazole and pantoprazole. So second thing, what you have to see, this should not be the individual case literature. Means the literature which you are presenting here, it should not be ICSR kind of literature, means a single patient case. It should be more than a patient case, because that won't be a part of your ICSR. If it is a single patient, it will be a part of your ICSR. But if it is more than one patient, then you could not have documented in the ICSR, and that kind of safety finding should come here. This you have to remember always. People will ask you as to which kind of articles you present in PBER in any of the sections, whether it is related to risk or whether it is related to section 11, whether it is related to medication error also, if you are off-label use that you are presenting. So all those things, if it is more than one patient, not a single individual patient case, then it will come under your aggregator.
So Amit, in our company, there are literatures where more than 1,000 or 2,000 patients are there, and we are splitting that literature into 1,000, 2,000, how many patients are there into individual case. Okay. Now it is in the ICSR database as well, and it is for more than one patient as well. How do we tackle this?
So, okay, tell me. So if you are splitting it into 1,000 cases, so see, this is the lacuna in the convention that you are doing. So are those valid cases, those 1,000 cases?
If the patient identifiers are not there, we'll proceed as a summary cases.
Summary cases. So in this case, what will happen? This is just a convention that you are doing. Ideally, we don't do it. That literature will automatically come under the PBER. And in case if you have also database in your ICSR, then also that case will come under this PBER literature section, if it is a significant finding for any of these criteria. So it will come under the PBER also. Not every client is split those cases in 1,000, 2,000, right?
Right.
It is from client to client. So that is a conservative approach that your client is taking. But that kind of literature will come under PBER. If it is more than one patient, it will definitely come under PBER. If it is having some significant finding.
Also, the signal literature. So this signal information will be in section 15 and 16.2.
If that is related to that specific risk, right? So that will not come under section 11. Like suppose drug-induced liver injury is your risk. Suppose important identified risk. And you got the literature of drug-induced liver injury. Then that will be part of that 16.3 under drug-induced liver injury. That will not come here, section 11. That is already a risk. Why it will come here, right?
No, 16.2 and 15. Section 15 and 15.
In that case, this will happen. In section 15, if you say, then in section 15, you won't find it. It will be there in 16.2. What is the condition? Like suppose a literature article was reviewed during your reporting period and it was found to be a signal. Then the signal was evaluated. Okay. And it was completed, means closed also. So in this case, it will come under section 11, section 15.1, section 16.2 also. If it is not closed, then it will come under section 15.1 and this is section 11.
Okay.
And also in the appendices of signal log, it will go. Because this is the trigger. This literature is itself a trigger for that signal, okay? Is it clear, literature section now? Literature is actually a very vast topic. So what we have to do in this, like there is a strength of evidence for literature. How do we identify strength? Also, we will take some time other day on that strength of evidence part. Next section, what is it? Section 12.
So Amit, this section 11, how to present data, we will take it from on any other day.
No, you have to present this data. You can't say that you are taking it from others. How do we present?
No,
Suppose
I'm saying like we will take this class on another day. How to present the data in this. section
No,
11.
no, literature section I have told you, as you present for that section 16.3 and all, na, in the same fashion you will present literature here. You will give just in 2 to 3 lines what was the objective of the literature and method in 2 to 3 lines. And then you will give what was the inclusion exclusion criteria in short, only 2 to 3 lines. And more focus will be on what was the result and conclusion and author comment from that. And based on that, you will identify all the 7-8 criteria that we discussed for causality assessment. Based on that, you will write your company causality. So this is how we present the summary of literature in this section.
Okay.
Okay. It is the same. Presentation is same as we do for other section. There is no difference. Only thing is that I am asking that we can discuss on some other day when we have time on some other day. We will discuss as to how we identify whether which literature is having more strength. That we will discuss on some day. Okay. Now section 12. It's fine literature. Let's move forward.
Yes.
Okay. So section 12 is other periodic reports, right? So what should come here?
Whichever other report we write.
What other reports?
Like,
Will we write signal report here?
Yes.
like radar, like any car for other reason. For Canada, we write car.
Your voice is very low. Can you be a bit loud?
Hello, Amit, like any other report, like for US, we write pattern. For
Will signal report come here?
single report means
Signal evaluation report, will it come here?
no, I haven't.
Any health authority query report will come here? Those are also periodic report that we are writing, right? Yes, no, maybe?
According to me, yes, it will go.
No, only periodic reports that we are writing, whether any PADER you are writing for this or any local region PBER you have written during this reporting period. Means, suppose your reporting period is 1st January 2024 to 31st December 2024. So it might happen that within this reporting period, you could have submitted a quarterly PADER, almost 2, 3 or 4 quarterly PADERs to US region. So all the 4 quarterly PADERs you will mention in tabular format. In this, what we do, we write report type like PADER. Then what was the reporting interval for that? Any significant finding from that? So this is the column that we present here. So suppose that PADER was written, so you will write that PADER here, that these PADERs were there were no significant safety findings, or if safety finding is there that you have observed, then you can write there. Mostly 99.99% cases, there is no significant safety finding from any of the periodic reports. Then if there, suppose you have written some local region like Canadian authority reports, if you have written for Canada, then you can write that here. So these kind of periodic reports, the similar to those that are related to the PBER, if you are writing something or some local region or something, then you can present here. Okay.
So we will provide the conclusion of those reports.
That will be in your database, na? Your tracker, your aggregate tracker. In that aggregate tracker, you can find all kind of reports. You have to fetch that data from there.
No, I'm saying like here in this section, whatever other reports are there, we will provide conclusions of those reports here.
See, every report has a section of conclusion, right? So in that conclusion, you can go and check whether there is any significant finding reported in that conclusion. If yes, then you can present as a significant safety finding. Otherwise, no significant safety finding was there. Correct?
Correct. So if let's say the example of combination product, there are 3 papers written. For one for combination, 2 for other individual molecules. So if it is a combination product paper, then for individual paper we also take the data and present it here.
If you are providing it in combination therapy, then you have to present it here also. It is a combination product, so you will provide individual molecule PBER data here. Okay. But not vice versa in this case. Suppose you are writing individual molecule case, means PBER, then you won't provide the combination product PBER in this section. Did you get it? Like you are writing this for, suppose, paracetamol. So whatever periodic reports you have written for paracetamol, that will come here. But you won't present the data for paracetamol and cetirizine combination here. But vice versa, if you see, if you're writing this paper for paracetamol cetirizine combination, in that case, you will present if any other periodic reports you have submitted for individual paracetamol and cetirizine.
Right, because if we are presenting any combination data here, it will include the other drugs data as well.
Right.
So that's why we are not adding it.
Got it? Clear? Other periodic reports?
Yes.
Turi kutu यहाँ है. ठीक है Turi. Turi.
Please mute your mic. Pushpak. Chalo, next section is, what is the next section? Next section, people tell me.
Lack of efficacy in control clinical trial.
Efficacy in? So will we present lack of efficacy cases here? Tell me.
Yes.
Yes, yes.
From post-marketing?
So no.
From clinical trials only, not post-marketing. Okay.
So why the word controlled is there? Because of clinical trial, anyways, it is a controlled study. Right?
Control means it should be sponsored by you. Simply. Okay. So only lack of efficacy cases from the controlled clinical trials or which you are a sponsor for that clinical trials that will come here. Okay. Like suppose if you are doing some clinical trial for some molecule for, let's say, multiple sclerosis molecule, clinical trial is ongoing for the indication multiple sclerosis. So if you find that there are the patients or subjects who are not responding to that, it is a lack of efficacy for them. So those kind of cases will come here and you will present it and the analysis format only. Most of the times in clinical trials, we don't find lack of efficacy cases. So it's you can say there was no lack of efficacy cases observed during controlling. If there is anything, then you will present whatever information you got.
But Amit, often we do receive the disease progression cases.
That is
in
not
the
lack of
clinical.
efficacy. That is not lack of efficacy. There would be convention in your client also. So you cannot code it to lack of efficacy. That is a disease progression because those are kind of diseases, the cancer and all, they are meant to be progressing. So those are not, you can say, lack of efficacy. Do you code them as lack of efficacy?
No, we have some other convention for coding lack of efficacy.
Right. So these are disease progression, not lack of efficacy. Okay. Is it clear? Lack of efficacy section, people? What do we present here in lack of efficacy?
Yes, Amit sir. And how we write about the format?
Format is nothing like suppose it's simple. Hardly one or two cases will be there. So you will present that these many cases were received for lack of efficacy during this clinical trials, and then you will present what was the course of the event, like how that event happened. Like you write the narrative for that. So in the same way, you will write a cumulative narratives that suppose you find three cases. So you will write that in these three cases, these many this age group was there. And then this was the medical history in this many case. In this case, this medical, in this case, this medical. The same cumulative narrative as we write. It is the same analysis that we present here. There is nothing specific, tabular or something format.
And the source will be CSR or what for this?
Source will be, you can say it, you could have databased into your ICSR, your clinical trial team, they could have presented it. CSR will become source, like suppose if it is finalized, then CSR could be the source. Or if an interim CSR is present, means presented or written, then that could be your source. Source could be any of those. Mostly clinical trials, we rely on their reports only. That is CSR and all those things.
Then
Sir,
for 11 and 12 section, what will be the source?
11 is open literature. Open literature means your literature team will provide you the literature that were screened, or you can say that came into during a reporting period. They will provide it in the Excel format, or some companies have a literature database from where you can put the reporting interval and they will give you the relevant literatures, so relevant for signal, relevant for aggregate. From those you can screen and then you can take your MR confirmation and present it here. For other periodic reports, your aggregate reporting schedule is there, from where you can get the information as to which aggregate reports for this molecule was scheduled during this reporting period. Then you can go to those reports and see if there is any significant conclusion provided for those reports. This way you can do that.
and I mean, sir, for lack of efficacy, we run line listing for it for our.
No, actually, that is a part of clinical trial teams. They provide us their CSR and everything is there. If there is anything database in your that you can say ICSR also, because this might happen that if there is lack of efficacy, then they will report to you and there will be in your ICSR database. So their study number will be there. So from that study number, you can check in your database SharePoint and find out what is the report written for them from them, and then you can take information from them.
So the PT will be only lack of efficacy.
Lack of efficacy means there could be minimal effect. Whatever lack of efficacy SMQ is there, you can see there is a narrow SMQ of lack of efficacy. So whatever PTs are there, that will be fetched into that line listing. We use lack of efficacy, narrow SMQ for that. Clear?
Yes, sir.
Yes.
Chala. Next section is, what is next section?
Late breaking information.
What will come here?
Whatever data, which significant data from DLP plus 30 days.
Significant safety data, okay. So I will write late breaking period is like everything that you are getting line listing, everything that is up till DLP. So it might happen that there could be some data that arises during late breaking. That's why client keep it as DLP plus 30 mostly.
And sir, from where we get this data?
From your database, safety database.
So any
Okay.
literature screening we will do at DLP plus 30 days.
Literature screening, yes, you will do. Literature screening is an ongoing process. You being an aggregate team, there is a literature team also that do screening. So they would be doing their screening process throughout the year. So in case if there is any DLP plus 30, so you can ask them post 30 days, you can ask them that please provide us the new line list, new literature articles that you have found during this reporting period. They will provide you, and from there you can identify whether there is any significant safety finding. So if it is from literature, then you will present in the same manner, summarize the literature as you present for all other sections, just objective aim, results, comments, author, and then author's comments, and then your company causality or company comment. So you will present in that format. If there is any significant safety finding in your safety database, you will present it like that. If there is any clinical trial safety finding, they will let you know, or if you have access to their clinical safety database, you can find from them. And there might be certain like RSI changes there due to some safety reasons or non-safety reasons, whatever is there. So you will come to know if your RA team will tell you that your RSI is updated by this time, or you can go to, we have access to the RSI SharePoint where we can go and check in the DLP plus 30 days whether RSI is getting updated, approved or not. or is it the same? So based on all this information, we provide here.
So any action taken due to safety reason.
Any action taken due to safety reasons in the late breaking period, or any regulatory authority update that you get in late breaking information, you can present all these things here. Whatever is whatever you come across post DLP.
Any signal is identified and
Anything,
completed.
any safety related issues that you can present here. Any significant safety issues, whether it is signal, whether it is a regulatory authority or health authority update, whether it is RSI update, whether it is any significant finding from your ICSRs or from literature you have, or whatever is there, any significant safety finding, if you find, you will present it here. If it is post DLP and within 30 days.
Amit sir, in short, means whenever we request for data, we request data with DLP plus 30 days only.
No, where you will request this data?
Means for line
At the
list.
starting itself?
No, afterwards.
No, at the time of DLP, like in DLP, by the day of DLP or something, ideally what happens, we don't request data for this. We have access to all those information. When we are working as an aggregate, we have access to a literature, that SharePoint. We have access to RSI SharePoint. We have access to health authority websites where we can search. That is the clinical trial only that we don't have most of the times access. We need to ask them if there is any, or the clinical trial author will endorse it. You can write a comment here in the late breaking information for clinical science team. Kindly confirm if there is any late breaking information received during this reporting. Means from suppose 31st December is a DLP. So you will write a comment in the document itself while initiating a collaborative authoring. Please present any significant safety data observed from 1st January 2025 to 30th January 2020. So they will respond to your comment. We don't request data for that.
So this is collaborative authoring, or we will write this section?
This is our responsibility. If it is a clinical part information, then they will provide you. Otherwise, all the other things is your responsibility. Any doubt till here, people? Yes, no, maybe?
So Amit, so here from section 6 till section 14 we have covered. So there is confusion on like how we will author or collaborative effort is there. For which section it is collaborative effort and what this section we will only author. So there is
The whole PBER you will initiate for collaborative authoring. Now, safety sections will be your responsibility. You have to populate and your MR or your safety lead has to endorse it. Okay. Other than safety sections, you will populate whatever information you have. Then the respective stakeholders will either confirm or endorse it. Okay. And if there is any comments, they will provide the comments. So post collaborative authoring means collaborative authoring is initiated for everybody, whosoever is responsible for each section. They will be there. And post that, whatever comments you receive during collaborative authoring, you will collate all the comments, resolve those, whatever you are able to resolve. If you are not able to resolve certain comments, you have schedule a meeting with them. You will resolve the comments, and then a clean copy you will initiate for core review. But before initiating a core review, all the comments once you have addressed, you will initiate that comments address copy for all the stakeholders who were there in collaborative authoring to confirm that all comments are addressed. Okay, so this is how the process follows. There is nothing like you are only responsible, you will solely write and keep it there. If you are writing the safety sections also, then also that has to be endorsed by your MR and your safety lead. So everything has to be done in collaboration only. We are just writing the base information, means whatever information is there, we are collating and we are writing it.
So Amit, can you provide a summary apart from section 15 and 16? What other safety sections we will have to write? Section number only will do.
So what other sections you think you should write? Based on the training that you did till now, what other sections do you think are the safety sections that you should write?
Okay, 15, 16, so we will write.
Start the PBER from first section. Which sections you are responsible? Tell me.
So first introduction we will write.
Correct.
Second, we will write it is like data from the RA team, we will get worldwide marketing authorization.
We will append it as
uh,
an appendix, but template we have to write in section 2. Okay.
Yes,
Chalo, next.
section 3, any action taken. So this is a safety related. So we will have to write next section.
Right.
So RSI section 4, we will have to write.
Okay.
And section 6, 1 second.
Section 5.
Section 5, sorry. That is sales data, exposure data that already provided to us. So we
No.
will just.
We will write.
No, we calculate, we write those. We get only units sold.
Oh, okay, units sold. Okay. And data in summary tabulation, that is a collaborative authoring.
Collaborative authoring or ready to append.
Yes, ready to append. Okay. Then section 7, it is also collaborative authoring.
Mainly clinical science team responsibility, you can say.
And section 8,
non-interventional team responsibility.
Yes.
But we write it and they just confirm it.
Section 9 also same. We write it.
Our responsibility.
Yes, our responsibility.
Correct. That is medication error. And
Other
then other
clinical trials.
trials. So all information, that is our responsibility.
Section 10 is also our responsibility.
What is section 10?
Non-clinical data.
No, that is non-clinical team section responsibility. They have to write it.
Okay, literature section 11, it is our responsibility.
Correct.
Okay. Other periodic reports also we will take from the database.
Our responsibility, not database, from the tracker and all those things you can see.
Yes, on those.
It's simply your responsibility.
Lack of efficacy, it is collaborative.
Collaborative, our responsibility to write, they will endorse it. Clinical science team will endorse it.
And lead breaking is our responsibility.
Our responsibility.
Section 15, 16, our responsibility.
Our responsibility. Section 17, 18 is also our responsibility. But in innovator molecules, if there are innovator molecules or innovator client, that is the medical affairs responsibility. So ideally, we should know as to what information should go in section 17, 18. If you are working for generic molecule, then definitely 17, 18 is your responsibility. But if you are working for innovator clients, then it is a medical affairs responsibility, but you should know as to what information goes in that. Okay. Am I audible?
Yes, Amit sir.
Yeah.
Yes, the conclusion we will write.
We will write. There is a template text. There is nothing change in conclusion. Don't worry. But MR confirms it, whether the conclusion, if there is any change or something. So in short, everything is our responsibility. There are only few sections that we rely on other people. Otherwise, PBER is your responsibility. Okay.
Also, summary, executive summary, we will have to write.
Executive summary, this is a copy paste. I'll tell you in last. You no need to write anything new. It is from the below sections only. You copy paste the information there. There is nothing new that you write here. Okay. Chalo, today I have less time. So we will continue in next session. Now we are pending with 17, 18 in PBER, right?
Yes.
So probably we will cover 17, 18 next, and then maybe a few more topics in some other sessions after that. So now you people are good to go ahead. Whatever 17, 18, you can say direct that it is medical affairs responsibility, and you can now face any of the interviews if you want. Okay. Chalo, shall we conclude the call today now?
Just last question, Amit. So
Yes, tell me.
throughout the PBER, we will use that which reference technique we use, that country name for appendices.
Country name means?
Like if we are to cite something, how we are doing citing and that format of citing.
No, literature you are meant to say? Literature citation?
Yeah, citation. So we will write it in the reference section, right? So that
Reference section
format.
will be there, and that Vancouver style of formatting.
Yes,
Vancouver style of referencing.
So throughout the paper, it will be the same format. Right?
Like suppose if you are written some thesis, then you will provide the numbers in the main report. Okay, 1, 2, 3, 4 in the sequence how the literature is being referenced. And if something is cross-referenced, there is a end note for literature referencing. Your client will give you in your Word, it will be there. So it is easy to refer, no problem with that. Just you write the name of the Vancouver style referencing, you cite from whatever you are taking. You have to put into the end note, it will take at the place where you are inserting it. So it will automatically number it 1, 2, 3, 4. So in main report body, it will be 1, 2, 3, 4 numbering. And in reference section, the last section, you will have the complete citation.
Okay, okay.
Okay. Chalo, then let's conclude today's call.
Okay, Amit. Thank you.
Chalo, thank you. Bye bye, everybody.
Thank you, Amit.